Hi, and welcome back to my channel, Learning Biology with Dr. Vanessa, where we take difficult biological concepts and make them easy to understand. In today's video, we are going to take a closer look into the urinary system by taking a look at the nephron, which was introduced in the last video, and I mentioned it is the functional unit of the kidney. Here we're going to really delve in and talk more about the nephron and how it works. I want you to imagine a water filter. It works by separating out impurities, at the same time keeping the clean water flowing. Now imagine a built-in filtration system inside your body working around the clock to clean your blood and remove waste. That's exactly what your kidneys do, and it all happens inside millions of microscopic units called nephrons. In the nephron, every drop of blood is carefully filtered, balanced, and waste is removed. But how does this tiny structure handle such a massive task? How does the nephron remove waste from the blood and turn it into urine? Let's take a closer look. Each nephron consists of two parts, a renal corpuscle and a renal tubule. The renal corpuscle houses the glomerulus, which is a ball of capillaries, and Bowman's capsule. This is where blood plasma filtration begins. And the renal tubules is where the filtered fluid passes. The renal tubule includes three parts: the proximal convoluted tubule, the loop of Henle, or nephron loop, and the distal convoluted tubule. Proximal refers to the part of the tubule that is attached to the renal corpuscle, and distal denotes the part of the tubule that is further away. The distal tubules of several nephrons empty into a single collecting duct. The collecting ducts unite and convert into papillary ducts, which drain into the minor calyces, then to the major calyces, the renal pelvis, and then out of the kidney through the ureter to the urinary bladder, as we discussed in the last video. Now that we have a basic understanding of how the filtrate enters and exits, let's go back and talk more about the anatomy of the nephron. The renal corpuscle houses the glomerulus. This is that ball of capillaries that brings blood into the nephron. This blood enters through the afferent arteriole and exits through the efferent arteriole. The glomerulus itself consists of fenestrated capillaries. Remember, those are capillaries that have bigger pore sizes, which allow water and small molecules to pass through more easily, but it does keep the proteins and blood cells inside the bloodstream. The fluid filtered from blood is now called glomerular filtrate and passes into Bowman's capsule. This capsule is a double-walled epithelial cup consisting of visceral and parietal layers. The visceral layer contains podocytes. These cells have many foot-like projections that wrap around the endothelial cells of the glomerular capillaries, and together they are going to form the inner wall of the capsule. The parietal layer consists of simple squamous epithelium, which allows easy filtration and forms the outer wall of the capsule. The filtered fluid, or glomerular filtrate, enters into the capsular space, which is the space between the two layers of the glomerular capsule, also known as Bowman's capsule. From here, this fluid is going to enter into the nephron to determine what will remain in the nephron and eventually exit the body as urine, or what the body needs and is necessary to be reabsorbed back into the body. From Bowman's capsule, the filtered fluid will pass into the proximal convoluted tubule. The proximal convoluted tubule is lined with cuboidal cells that have microvilli. This increases the surface area for reabsorption because in the proximal convoluted tubule, most reabsorption is going to happen. Moving forward, we are going to reach the nephron loop, or loop of Henle, and this loop contains both a descending limb and an ascending limb, which is going to create a concentration gradient to regulate water and salt balance. About 80 to 85% of the nephrons in the kidney are cortical nephrons, which means that their renal corpuscles lie in the outer portion of the renal cortex. These nephrons have short nephron loops that are found mainly in the renal cortex and just slightly enter into the medulla. These short nephron loops receive their blood supply from the peritubular capillaries that come from the efferent glomerular arterioles. The other 15 to 20% of nephrons are juxtamedullary nephrons. These nephrons have their renal corpuscles deep within the renal cortex, very close to the medulla, and they have long nephron loops that extend into the deepest region of the medulla. These nephrons receive their blood supply from the peritubular capillaries and the vasa recta that come from the efferent glomerular arteriole. Nephrons that have long nephron loops are responsible for the kidney's ability to excrete very dilute or very concentrated urine, a topic we will talk about more in detail in a later video. The descending limb and the thin portion of the ascending limb are composed of simple squamous epithelium. The thick ascending limb is composed of simple cuboidal to low columnar epithelium. In each nephron, the final part of the ascending limb of the loop of Henle makes contact with the afferent glomerular arteriole that enters into the renal corpuscle. In this area, the columnar cells of the ascending limb are crowded together and are known as the macula densa. In the area of the afferent arteriole that connects with this portion of the ascending limb, there are modified smooth muscle fibers called juxtaglomerular cells. Together, the juxtaglomerular cells and the macula densa make up the juxtaglomerular apparatus. This apparatus helps regulate blood pressure within the kidneys, which we will discuss in a later video. The distal convoluted tubule begins just after the macula densa. In the distal convoluted tubule, we find principal cells, which have receptors for both antidiuretic hormone and aldosterone. These hormones will regulate the functions of the distal convoluted tubule and the collecting ducts. There are also a lesser number of intercalated cells which play a role in the homeostasis of blood pH. We also see these types of cells in the collecting ducts. Many distal convoluted tubules will connect to one collecting duct to drain filtrate into. The distal convoluted tubule will work to fine-tune electrolyte levels, and the collecting duct will determine the final concentration of urine. The nephron carries out 3 essential processes. First is glomerular filtration. In glomerular filtration, blood is going to enter into the glomerulus under high pressure, which is the pressure that is driven by normal blood pressure. Fenestrated capillaries, remember those that have bigger pore sizes, and the filtration membrane are going to allow water, ions, glucose, and waste to pass into the Bowman's capsule, forming filtrate. Large molecules like proteins and blood cells are retained in the bloodstream. This process is driven by glomerular filtration rate, which must stay in balance for the kidneys to have proper function. Next would be tubular reabsorption. In the proximal tubule, about 99% of filtered water and many useful solutes like sodium, glucose, and amino acids are actively reabsorbed into the bloodstream. The loop of Henle plays a key role in water conservation by creating a salt gradient in the medulla of the kidney. And the distal convoluted tubule and collecting duct are responsible for fine-tuning reabsorption, and this is controlled by hormones like aldosterone and ADH. These hormones help the nephron adjust to the body's needs, whether that means retaining water when dehydrated or eliminating excess salts when necessary. I want you to take a note that reabsorption refers to returning these substances back to the bloodstream. The term absorption, which we talked about in the digestive system, means the entry of new substances into the body. And then finally, the nephrons participate in tubular secretion. This is where the nephrons actively secrete waste, drugs, and excess ions like hydrogen ions and potassium ions into the tubules to regulate pH balance and eliminate waste. This ensures that harmful substances like drugs and metabolic byproducts are efficiently removed from the body. Another note that secretion refers to removing a substance from the blood and eliminating it from the body. Anything that stays in the nephron and moves past the collecting ducts and enters into the minor calyces through the papillary ducts will be removed from the body as urine. Without properly functioning nephrons, waste and excess fluids would build up in the body, leading to toxicity, high blood pressure, and kidney failure. To keep your nephrons working at their best, stay hydrated, eat a balanced diet, and avoid excessive salt and toxins. In this video, we've laid the foundation by exploring the anatomy of the nephrons and touching on the basics of renal physiology. But there's so much more about renal physiology that we're going to be talking about, and that's exactly what we'll be doing in the next video. If you enjoyed this video, don't forget to like, share, and subscribe so that you never miss out on new content. Stay tuned, and I'll see you in the next video.